Warm welcome to all. I am Golda Albert, working as a tutor in Sri College of Nursing, Department of Fundamentals of Nursing. I am going to present sterilization. It comes under microbiology. Let me see. The process of killing or removing bacteria and all other forms of living microorganisms and their spats from preparation. Essential concept in the preparation of sterile pharmaceutical products. It means free from microorganism. Let me see the aim. To provide a product that is safe and eliminates the possibility of introducing. And then importance of sterilization to prevent contamination in sterile products. To prevent transmission of pathogenic microorganisms which are responsible for causing disease in plants, animals and human beings. To prevent decomposition and spoilage of food and food products. To prevent the contamination of unwanted microbes in pure cultures and other microbiology experiments performed for research studies. To prevent unwanted microbial contamination in antibiotic, enzyme, vitamins, fermentation and other industries process. To prevent contamination in aseptic areas, instruments which are used for the preparation of sterile dosage forms and sterility testing. And then why we need sterilization? Microorganisms capable of causing infection are constantly present in the external environment and on the human body. External environment means it's all over the area. And then microorganisms are responsible for contamination and infection. The aim of sterilization is to remove or destroy the microorganism from materials or from surfaces. It means free from microorganism. And then let me see the definition of important terms. First of all, we are going to see the sterilization. It is a process by which an article, surface, or medium is made free of all microorganisms, either in vegetative or spores form. And then disinfection. It is a process of destruction of all pathogens or organisms capable of producing infection in living cells, but not necessarily spores. All organisms may not be killed, but the number is reduced to a level that is no longer harmful to health. Then disinfections. These are antimicrobial agents that are applied to the surface of non-living objects to destroy microorganisms that are living on the objects. We are all using in this at the home. Antiseptics, chemical which can safely be applied to living tissues and are used to prevent infection by inhibiting the growth of microorganisms. And then asepsis, technique by which can occurrence of infection into an uninfected tissue is prevented. And then bacterial agents or germicides. These are the chemical substances which are able to kill bacteria and then germs. Let we see the methods of sterilization. There are two methods are there. First one physical method and then second one chemical method. In physical method under that dry heat, moist heat, radiation and then filtration and mechanical method. In dry heat sterilization under that for an example incineration, direct flame, red heat and hot air. And then in under that moist heat for an example pasteurization, denaldization and then autoclave. Under that sterilization by radiation for an example use of ultraviolet rays like UV light, non-ionizing and ionizing radiation, X-rays, gamma rays and then beta rays. In filtration or mechanical method for an example aspartus filter, sinter glass filter, filter candles and then membrane filter. And then second one chemical method. It's under that gaseous sterilization and sterilization by disinfectant. In gaseous sterilization for an example ethylene oxide gas, formaldehyde and beta propiolactone. In sterilization by disinfection under that alcohols and aldehyde, phenyl and halogens, and oxidizing agents, and then salt. We are going to see differentiated between antiseptics and disinfection. First of all, antiseptic used for humans and animals. In disinfectants used for non-living things, furniture and other household items. Household items, benches, chairs, all over the things. And an antiseptic commonly found in healthcare centers or hospital. In disinfectant, commonly found in homes or public places. In antiseptic, cleans wound and surgical site to prevent infection and other complication. In disinfectants, kills microorganisms on the surface of non-living things. In antiseptics, includes mouthwash and cold sore and yeast infection treatment creams. In disinfectants, includes cleaning products for houses and public places. In antiseptic, transport through the lymphatic system and destroyed bacteria within the human body. And disinfectants destroy the cell wall of microorganism or interferes with the metabolism of microbes thriving on the surface of dangeable objects. In antiseptic, not harmful to humans and then animals. In disinfectants, harmful to humans and then animals. And then, how can microorganisms be killed? Denaturation of proteins, interference with protein synthesis, interruption of DNA synthesis or repair, oxidative damage of cell, and disruption of cell membranes. And then factors that influence efficacy of disinfection or sterilization. First one, contact time. Second one, physico-chemical environment. For an example, pH level. And third one, presence of organic material. And then fourth one, temperature. Fifth one, type of microorganism. And the number of microorganism. Last one, material composition. What to sterilize? All instruments that penetrate soft tissues and bones. Instruments that are not intended to penetrate the tissue, but they may come into contact with oral tissues. If the sterilization procedure may damage the instruments, then sterilization can be replaced by disinfection procedure. Let me see the methods of sterilization. First one, physical method. Involves process by the use of physical means, utilization of heat in the presence or in the absence of heat, moisture, radiation, or membrane filtration method. Let me see the physical methods of sterilization. If we are maintaining dry heat means we are using in the oven. In moist heat means we are using autoclave. In radiation means we are using gamma ray chamber. And then dry heat sterilization. Here this is the most reliable and rapid method of sterilization. In mechanism, protein denaturation, oxidative damage, and toxic effect of elevated levels of electrolytes. Time required for sterilization is inversely proportional to the temperature of exposure. This can be exposed as thermal death time, which is the minimum time required to kill a suspension of microorganism at a temperature and specific condition. First of all, we are going to see sunlight and drying. In household method, we are using this. Action primarily due to UV rays, which means ultraviolet. However, effects vary due to places. For an example, natural method for sterilization of water in tanks, reservoir, lakes, and then etc. Second one, heat. Most reliable methods of sterilization and should be the methods of choice. For an example, inoculating wire, needles, and forceps, and then etc. And then third one, flaming. Passed over flame without allowing it to become red hot. Most of all, we are in this method using in the laboratory department. For an example, culture tube, glass slides, scalpels, needles, cover slips, and then etc. Fourth one, incineration. Excellent method for rapid destroying materials. For an example, pathological material, contaminated cloth, animals carcasses. And then hot air oven. Hot air ovens are electrical device used in the sterilization. The oven used dry heat to sterilize articles. Generally, they can be operated from 50 to 300 degrees centigrade. It means 120 to 572 degree Fahrenheit. There is a thermostat controlling the temperature. There is a most widely used methods of sterilization by dry heat. In items, glassware, forceps, scissors, scalpels, all glass syringes, swabs, liquid paraffin, dusting powder, fats and then grease. It is not suitable for surgical dressing and rubber items, plastic, volatile and heat liable substances. In this, instruments look like a oven, micro oven. Let me see the precautions. Glassware should be dry. Oven should not be overloaded. Glass material after drying are allowed to cool down before use because of preventing the breakage. Articles not to be arranged in a manner to allow free circulation of air. Door of the oven should be open after it cool down. And then we have to maintain some temperature and then timing. In we are maintaining 170 degree centigrade means in we are maintaining 60 minutes. In 160 degree centigrade means maintaining 120 minutes. 150 degree centigrade means we are maintaining 150 minutes. In 140 degree centigrade means we are maintaining 180 minutes. Normally, the spores as well as the vegetative forms of all microorganisms are killed in 2 hours at a temperature of 160 degrees centigrade. Let me see the advantages and then disadvantages. First of all, we are going to see about the advantages. It's very, very important. It is suitable methods of sterilization of substances destroyed by moisture. They do not require water and there is not much pressure buildup within the oven, unlike an autoclave, making them safer to work with. Suitable and easy to be used in laboratory environment, they are much smaller than autoclaves but can still as effective. Let me see the disadvantages. Long heating time and high temperature. As they use dry heat instead of moist heat, some organisms like prions may not be killed by them every time. And then moist heat sterilization. Killing of microorganisms with hot water or steam. In mechanism, denaturation and coagulation of proteins divided into three forms in terms of temperature. First one, temperature below 100 degrees centigrade. It means pasteurization method. And second one, temperature at 100 degrees centigrade. It means denatalization method. And third one, temperature above 100 degrees centigrade. It means autoclaving method. Let me see. Temperature below 100 degree pasteurization method used for milk, ice cream, yogurt, and fruit juices. Heat tolerant microbes survive. Batch method, temperature below 100 degree pasteurization of milk. Developed by Louis Pasteur to prevent the spoilage of beverages. Used to reduce microbes responsible for spoilage of beer, milk, wine, juices, and then etc. Milk was exposed to 65 degrees centigrade for 30 minutes. In inspissation is a process used when heating high protein containing media, for an example, to enable recovery for bacteria for testing. In high temperature short time pasteurization used today. Milk is exposed to 72 degrees centigrade for 15 seconds and target all non-sporing pathogens. For an example, Mycobacteria, Brucella, Salmonella, relatively heat resistant, may survive the holder method. And then second one, temperature at 100 degrees centigrade. In boiling method, boiling for 10 to 30 minutes may kill most of vegetative forms, but spores withstand boiling. Denatalization. Steam at 100 degrees centigrade for 20 minutes on 3 successive days. Used for egg, serum, and sugar containing media. Steam sterilizer. Steam at 100 degrees centigrade for 19 minutes used for media which are decomposed at high temperature. And then third one, a temperature above 100 degrees centigrade. In that autoclaving method, steam above 100 degrees centigrade has a better cooling power than dry heat. Bacteria are more susceptible to moist heat. In components of autoclave consist of vertical or horizontal cylinder of gunmetal or stainless steel. Lid is fastened by screw clamps and rendered airtight by an asbestos washer. Lid bears a discharge stab for air and steam, a pressure gauge and a safety valve. And then precautions should not be overloaded and arranged in a manner which allows free circular of air. And third one, material to be sterilized should be perfectly dry, and test tubes, flask, etc. should be fitted with cotton blocks, and perishables and peptides should be wrapped in paper. Rubber materials and inflammable materials should not be kept inside. The oven must be allowed to cool for 2 hours before opening, since glassware may crack by sudden cooling. Let me see the procedure. Water is added on the bottom of the autoclave and articles to be sterilized or placed in the perforated shelf. The lid is closed, discharge tap is open, and safety valve is adjusted to the record pressure. When the air bubbles stop emitting from the discharge tap, it indicates all the air from inside the autoclave has been removed. At this stage, the discharge tap is closed. Steam pressure rises inside, and when it reaches the desired set level, the safety valve opens and excess steam escapes. From this point, the holding time 15 minutes is counted. When the holding time is over, the heating is stopped and the autoclave is allowed to cool. Chill pressure gauge indicates that the inside pressure has reached to the atmospheric pressure. The discharge tap is opened slowly and air is allowed to remove from the autoclave. The lid is open and the sterilized articles are removed. Record temperature and time. We are maintaining correct time and then temperature. It's must. In temperature, we are maintaining 121 degree centigrade means in the duration time it's 50 minutes. 126 degree centigrade means duration time is 10 minutes. And 130 degree centigrade means duration time is 3 minutes. And then uses of autoclaves. Useful for materials which cannot withstand high temperature. To sterilize culture media, heat stable liquids, saline solution, heat resistant equipments and instruments, glasswares, ampoules, syringes, rubber material, gowns, surgical dressings, gloves, and etc. Unsuitable for anhydrous materials such as powders, oils, fats, and then ointments. More efficient than dry heat sterilization. First one, it provides greater lethal action of moist heats. And then second one, it is quicker and heat up the exposed articles. It can penetrate easily porous materials such as cotton wool stoppers, paper and cloth wrappers. And then third one, radiation. Type of radiation, ionizing radiation and non-ionizing radiation. First one, non-ionizing radiation. It's a hot sterilization method. Infrared is for rapid mass sterilization of pre-packed items such as syringes, then catheters. And then ultraviolet used for disinfecting enclosed areas such as entryways, operation theaters, and then laboratory. And then second one, ionizing radiation. It's a cold sterilization method. Gamma rays is for X-rays used for sterilizing plastics, syringes, swabs, catheters, animal feeds, and cardboard, oils, greases, and fabric and metal foils. And then final one, filtration method or mechanical method. It helps to remove bacteria from heat level liquids in items, sera and solution of sugars and or antibiotics. In principles, as viruses pass through the ordinary filters, filtration can be used to obtain bacteria free filtrates of clinical samples of virus isolation. And type of filters: candle filters, asbestos filters, sintered glass filters, and membrane filters. Let me see one by one. First of all, candle filter. Manufactured in different grades of porosity and widely used for purification of water for industrial and drinking purpose. Made up of porous porcelain, inexpensive and available in different sizes. An asbestos filter. It's disposable, single-use disc made up of asbestos. Tend to alkalinize filter liquids. The pore size of filters ranges from 0.01 to 5 microns. Usage is discouraged because of its carcinogenic property. And then next, sintered glass filter has low absorption properties. Borosilicate glasses finally powdered in a ball mill and packed into disc mold and heated until suitable adhesion take place between the granules. Bridal and expensive. Let me see the membrane filter or ultra filter. Made of cellulose esters or other polymers, usually used for water purification and analysis, sterilization and sterility testing, and preparation of solution for parenteral use. And then the, there are 150 micro thick and containing millions of microscopic spores ranging from 0.01 to 10 micro in diameter. Let me see the advantages and then disadvantages. First of all, advantages. All microorganisms are separated by process of sheathing. Membranes have a high and uniform porosity, permitting a rapid rate of filtration. Membranes are disposable, hence there is no cross contamination between filter products. Absorption is very less. And the disadvantages, pre-filter is used before the membrane filter to avoid clogging and breaking. They have less chemical resistance to certain organic solvent such as chloroform, ketone, and then esters. Let me see the chemical methods of sterilization. Action of chemical method in mechanism: protein coagulation, disruption of cell membrane resulting in exposure, damage, or loss of contents. Removal of sulfitryl group essential for normal functioning of enzyme. Substrate competition. Commonly used chemicals. First one, reagents. And under that, alcohol, aldehyde. In alcohol, frequently used for ethyl alcohol, isopropyl alcohol. This must be used at concentration 60 to 90 percent. In isopropyl alcohol is used in disinfection of clinical thermometer. Methyl alcohol is effective against fungal forts, treating cabinets and incubators. And methyl alcohol is also toxic and inflammable. In aldehyde, formaldehyde having bactericidal, sporicidal, and has lethal effect on viruses. Used to preserve anatomical specimen destroying anthrax, spores on hair and wool. And then glutaldehyde, third one, glutaldehyde. Effective against tuberculosis, bacilli, fungi, and then viruses. Less toxic and irritant to eyes and then skin. Used to treat anesthetic rubber, face mask, plastic endotracheal tubes, and metal instruments and polythene tubing. And then second one, dyes. There are two dyes are there, aniline dye and acrylidine dye. Both are bacteriostatic in high dilution but are of low bactericidal activity. An aniline dye is more active against gram positive than the gram negative organisms. Some important dyes proflavin, agriflavin, euflavin, and aminoglycine. This impair the DNA complexes of the organism and thus kill or destroy the reproductive capacity of the cell. Halogen in under that iodine and chlorine. In iodine used as skin disinfectant, having active bactericidal activity and moderate acting on spores. In chlorine used to disinfect water supplies and swimming pools and food and dairy industries, along with hypochlorites or bactericidal. Also act on viruses. And then penols. These are obtained from distillation of coal tar between 170 to 270 degrees centigrade. Lethal effects are capacity to cause cell membrane damage, releasing cell contents and causing lysis. Low concentrate will precipitate proteins. And then third one, gases. Types of gases used for sterilization: ethylene oxide, formaldehyde gas, and beta-propiolactone. In ethylene oxide, action is due to its alkylating the amino, carboxyl, hydroxyl, and sulfhydryl groups in protein molecules, also on DNA and RNA. In items, hot lung machines, respirators, sutures, dental equipments, books, and then clothing. Let me see the formaldehyde gas. This is widely employed for fumigation of operation theatre and other rooms. Other room means it may be a ward, all ICUs. And then formaldehyde is produced by adding 150 gram of KMNO4 to 280 ml of formalin for every 1000 square feet of room volume. After closing the windows and other outlets, after fumigation, the door should be sealed and left unopened for 48 hours. It's really and very effective. Beta-propylactone, product of ketone and formaldehyde with a boiling point of 163 degrees centigrade, having rapid bactericidal activity but carcinogenic, capable of killing all microorganisms and is very active against viruses. Sensitive of microorganism. Microorganism shows resistant to heat, radiation, and then chemicals. The vegetative forms of bacteria and fungi are most sensitive. The thermophilic bacteria, smaller viruses, and mold spores are killed at temperature between 70 to 90 degrees Celsius, while bacteria spores may be destroyed at 90 to 120 degrees centigrade temperature. Sterility indicators. Changing appearance in color or pattern, the sterilization indicators visually show if cleaning conditions are passing or procedures have been completed. Eliminating any confusion or possibility instruments will not be sterile. Indicators are used routinely in clinical and research environment where contamination elimination is crucial. With the temperature resistance required to ensure the purification, the sterilization indicators are available in different forms such as tapes, ampoules, and then sticks. Monitoring of sterilization process can be achieved by the use of physical, chemical, or biological indicators. Let me see the physical indicators. First one, moist heat indicator. A moisture process record, it means MPR, is prepared as part of the validation procedure for a particular autoclave. The MPR should be checked at annual intervals and whenever significant changes occurrence in the PPR, it means batch production records. When compared with the MPR, microprocessor control Sterilization cycles are now a part of modern autoclaves. And second one, dry heat. In dry sterilization process, a temperature record chart is made of each sterilization cycle and is compared against a master temperature record. And third one, radio sterilization. A plastic dosimeter gives a accurate measures of the radiation that is absorbed and is considered to be the best technique and currently available for the radio sterilization process. And gaseous method. For gaseous sterilization procedure, elevated temperatures are monitored for each sterilization cycle by temperature probes and routine leak tests are performed to ensure gas tight seals. Gas concentration is measured independently of pressure rise, often by reference to the weight gas used. Pressure and humidity measurements are recorded. In fifth one, infiltration. Bubble point pressure test is a technique employed for determining the pore size of filters and may also be used to check the integrity of certain types of filters devices immediately after use. The principles of the is that a filter is soaked in appropriate fluid and pressure is applied to the filter. The presence difference when the first bubble of air breaks away from the filter is equivalent to the maximum pore space. When the air pressure is further increased slowly, there is general eruption of bubbles over the entire surfaces. The pressure difference is equivalent to the mean pore size. And then second one, chemical indicators. Chemical monitoring of a sterilization processes based on the ability of heat, steam, sterility gases, and ionizing radiation to alter the chemical or physical characteristic of a variety of chemical substances. Brown stew, most commonly used chemical indicators for heat process, contain small sealed colored tubes having a reaction mixture and the indicator. Exposure to high temperature resulting in the change of color of the indicators. It means red to turn over the green. And then second one, witness tube, consists of single crystalline substance of known melting point containing a glass tube. A dye may be included to show more clearly that the crystal have melted. Indicates that the certain temperature has been reached. And then third one, biological indicator, consists of a suitable organism deposited on a carrier and are disrupted through the sterilization load. At the end of the sterilization process, the units are recovered and cultured to determine the presence or absence of survivors. Confirm the ability of the sterilization process to kill microbial spores. Can check large number of spores. Includes all the parameters of the sterilization process. Most critical test of sterilization process.